the molecular copy machine. In the history of molecular biology, few techniques have altered the landscape of research as profoundly as the polymerase chain reaction, or PCR. Developed in the 1980s, PCR transitioned DNA replication from a complex in vivo process within living cells to a controlled in vitro enzymatic assay. At its core, PCR is a molecular photocopy machine. It allows us to take a negligible amount of genetic material and amplify a specific target sequence into a workable concentration. To understand PCR is to understand the balance of thermodynamics and enzymatic kinetics. Today, we will move beyond the basic definitions and examine the mechanistic why behind the three-step cycle. The master mix, a biochemical foundation. Before a single cycle begins, we must assemble a master mix. This solution contains several critical components. Each plays a distinct biochemical role. First, we have the template DNA. This contains the region of interest. However, the template is often a small fraction of a complex genomic mix. Next, we include primers. These are short, single-stranded synthetic oligonucleotides. They provide the 3' hydroxyl group necessary for DNA polymerase to initiate synthesis. We also add DNTPs, deoxynucleotide triphosphates. These are the building blocks, A, T, C, and G. They provide both the material for the new strand and the chemical energy required for polymerization. The buffer maintains a stable pH, which is essential for enzyme stability. Within this buffer, the most critical component is often magnesium chloride. The magnesium ion acts as a divalent cation cofactor for the DNA polymerase. It serves two roles. It coordinates the phosphate groups on the DNTPs, facilitating the nucleophilic attack by the 3' hydroxyl group of the primer. It also helps stabilize the negative charge of the DNA backbone. Without sufficient magnesium ion, the enzyme remains inactive. However, excessive magnesium ion can stabilize nonspecific primer binding, leading to experimental artifacts. Finally, we have the thermostable DNA polymerase, most commonly Taq polymerase. Unlike human polymerases, Taq can withstand the high temperatures required to melt DNA without denaturing its own protein structure. Step 1. Denaturation and thermodynamic reasoning. The first step of the cycle is denaturation, typically occurring between 94 and 98. From a thermodynamic perspective, DNA is held together by hydrogen bonds between complementary bases and base stacking interactions. At room temperature, the double-stranded state is energetically favorable because the enthalpy of the hydrogen bonds outweighs the entropy of single-stranded DNA. When we raise the temperature, we provide enough thermal energy to overcome these non-covalent bonds. The strands separate, or melt. It is important to note that the covalent phosphodiester bonds of the DNA backbone are much stronger and remain intact at these temperatures. This creates a population of single-stranded templates ready for the next phase. Step 2. Annealing and the melting temperature. Once the strands are separated, the temperature is lowered, usually to between 50 degrees centigrade and 65 degrees centigrade. This is the annealing step. The goal here is for primers to bind to their complementary sequences on the template. This process is governed by the melting temperature. The melting temperature is defined as the temperature at which 50% of the primer template duplexes are formed. If the annealing temperature is too high, the kinetic energy is too great, and the primers cannot form stable hydrogen bonds with the template. If the annealing temperature is too low, the primers may bind to sequences that are only partially complementary. This results in non-specific amplification. Researchers must carefully calculate the annealing temperature based on the GC content of the primers. Because GC pairs share 3 hydrogen bonds compared to the 2 bonds in AT pairs, sequences with higher GC content require higher temperatures to anneal specifically. Step 3. Extension and enzymatic synthesis. The third step is extension, usually performed at 72 degrees centigrade. This is the optimal temperature for Taq polymerase activity. During extension, the polymerase recognizes the primer template complex. It moves along the template in the 3' to 5' direction, synthesizing a new complementary strand in the 5' to 3' direction. The rate of synthesis for Taq is approximately 1,000 base pairs per minute. Therefore, the duration of this step is determined by the length of the target sequence. By the end of this step, we have 2 double-stranded DNA molecules for every one we started with. Why exponential amplification occurs? The power of PCR lies in its mathematical progression. Because the products of one cycle become the templates for the next, the DNA concentration increases exponentially. The theoretical yield can be expressed by the formula y equals a into 2 raised to power n, where a is the initial number of template copies, and n is the number of cycles. After 30 cycles, a single DNA molecule can, in theory, be amplified into over 1 billion copies, which is 2 raised to power 30. In practice, the efficiency is rarely 100%. As the cycles progress, DNTPs are consumed, the enzyme loses activity, and the high concentration of product can lead to self-annealing. This eventually leads to a plateau phase where no further amplification is shooting, the challenge of non-specificity. Even with careful planning, experiments can fail. A common pitfall for early career researchers is the appearance of primer dimers or non-specific bands on an agarose gel. Primer dimers occur when primers have complementarity to each other rather than the template. They are often visible as small, fuzzy bands at the bottom of the gel. They represent a significant problem because they consume DNTPs and enzyme resources. If you observe non-specific bands, the first variable to investigate is the annealing temperature. Raising the annealing temperature by 1 degree centigrade or 2 degrees centigrade often increases stringency, ensuring that only perfectly matched primer-template pairs are stable enough to initiate extension. Real-world application, RT-PCR in diagnostics. PCR is not limited to DNA. In clinical diagnostics, we frequently use reverse transcription PCR, RT-PCR, to detect RNA viruses, such as SARS-CoV-2. In this application, an enzyme called reverse transcriptase first converts the viral RNA into complementary DNA, cDNA. This cDNA then serves as the template for a standard PCR reaction. This allows clinicians to detect the presence of a virus with extreme sensitivity, even when the viral load is. And conclusion. To summarize, the master mix provides the necessary chemical environment, with magnesium ions serving as a vital cofactor. Denaturation uses heat to overcome the enthalpy of hydrogen bonding. Annealing is a delicate balance of temperature and specificity. Extension utilizes the kinetic optimality of Taq polymerase. The result is an exponential increase in target DNA. By mastering these variables, you gain the ability to manipulate the very blueprint. To analyze my results, I find myself thinking about the first cycle. In those first few minutes of the reaction, we are essentially asking a few strands of DNA to find their match in a sea of billions of other base pairs. It is a statistical needle in a haystack. I often wonder, how does the precision of the annealing temperature compensate for the sheer chaos of molecular motion? Is our calculated $T_M$ truly reflective of what happens inside that 0.2 milliliters tube? Or is it just our best mathematical approximation of a much more fluid reality? We treat these protocols as rigid laws, but in the lab, they feel more like a conversation with the laws of physics, one where the DNA sometimes talks back in the form of a smeared gel or an unexpected band. It reminds me that even our most standard tools are built on a foundation of profound molecular complexity.